everyone. My name is Rita Giuffrida, and I am a researcher at Trust IT Services, and I have the privilege of being one of the BioDT project members. In particular, I'm involved in communication and dissemination activities, and I am very pleased to welcome you all today for our second webinar that will focus on the use cases, so the BioDT use cases that are demonstrators that our team is building to test the biodiversity digital twin prototype. Today, we are going to present you how the use cases are going to be built. And in particular, the presentation will be divided in three main parts. In the first part of our webinar, you can find an interactive session that I will show you in a couple of minutes, where we can also understand a little bit better who you are. And after that, instead, I will pass the floor to Doug Andresen, who is instead one of the BioDT use cases co-leader. After that, you can instead hear a little bit more about our use cases. Actually, you can hear more about the group 2, 3, and 4 that we are developing. And at the very end of this, we will actually have another interactive session where you can also ask any questions you might be, let's say, curious to learn, to hear more. In particular, something that I would like also to ask you is that in this Zoom meeting, you can also find a Q&A section. So please, whenever you have any questions, feel free to use it. And because actually we can also reply either live or after during the dedicated sessions at the very end of the webinar. So hopefully you can also see my presentation, and I will just go through the interactive session on Mentimeter. If this is the first time that you're using this tool, actually what you have to do is to go through menti.com, so the link that you find here, and then you should be able, let's say, also one screen, the one that I am sharing right now. Okay. So whenever you go also to menti.com, press this code, so 36845958, and then you should be redirected exactly to this page. I can also copy this link to everyone so that you can also. Okay, good. I see that some people are already joining. Good. Okay, so we have people from Europe mainly. Okay, also a person from Cairo, which is nice. And it is actually very nice to see how many different cities we are joining, because our goal is also to make sure that our research results are spread as much as possible, because actually the research does not have any barrier. So yeah, I'm very pleased to see how many people we have today, and the number is just growing, so which is very nice. Okay. Now I will really share it just for another couple of seconds, and then I will show you the next question. Okay, by the way, I see that the number 3 are Helsinki, Edinburgh, and Stockholm, and Amsterdam. Let's say, welcome everyone again. So in the second slide instead, we would like to also ask, let's say, another personal question, if we may, that is, what is the field in which you work? So we would like to know if you have, okay, one has included the code for Mentimeter, which obviously is not the field, but yeah, because in this way we can also learn each other a little bit more and see if you have also some connection. I will work with the topics or the use cases that we are going to present today, which seems so because you also work in the ecology, in IT biodiversity, which is nice. And finally, also let's say we would like to know why you have decided to join this webinar. Just, I mean, will it be just out of curiosity or because you are an expert in this field? Feel free also to type down, to learn stuff, which is good. To find out about BioDT, which is nice. By the way, webinar that we are organizing today is the second of let's say, public events that we have organized. So if you would like also to learn something more about BioDT, we also invite you to go through our webpage and also watch the recording of our first webinar that we did in July. And it was really about a general, and there you can really see a general overview of BioDT, which seemed to be one of the reasons why, the main reason why people have joined today. So with this, I would like to thank you again, and then I will pass the floor to Doug instead, that will give you a first overview of the BioDT use cases. Thank you, everyone. And Doug, the floor is yours now.
Thank you. This webinar is to present the use cases. The use cases are designed to test the biodiversity twin fitness for scientific research, and they will help to develop, test, and implement this prototype that we are building in this project. And we will also, through the use cases, find the data sets we need for each of these tests, and also collect the best practices and transfer them to researchers, so teach them how to implement the use cases in their own research through training activities later on. The use cases are divided in 4 groups, and we'll hear from some of these groups today. So it is the species response to environmental change, the genetically detected biodiversity, dynamics and threats from and for species of policy concern, species interactions with each other and with humans. And today we'll hear from one of the use cases under the genetically detected biodiversity, one under the species of policy concern, the invasive species, and then the use case we have on pollinators. So we will start by hearing from Desalegn Chala. Are you ready?
Yes, I am ready. Thanks, Doug, for the introduction, and welcome everyone. I'll talk about the use case, which is related to genetically detected biodiversity, with a specific focus on crop-wide relatives. And there are 2 main factors which are affecting food security. The first one is population growth and followed anthropogenic impacts. And the second one is climate change. And human population has increased from 1 to 7 billion just over the past 200 years. And that is about 700% increment as compared to the number we have achieved since the time of our evolution. And this number is expected to reach 11 billion by the end of this century. But so the population density is mainly happening in developed countries, but agricultural production is decreasing by 2% per decade to meet the Sustainable Development Goal target by 2030, which is zero hunger. We need to boost our food production by 70%. And the drastic decrease in productivity is mainly occurring in highly densely populated areas. And because of that, we need crops with higher yield, with higher nutritive values, and the crops which are adapted to changing environment. And we have untapped genetic diversity in traditional cultivars, in land races, and wild crop relatives. And if you consider the crops which we are currently using, they are faced induced selection pressure by human being. For example, this is the wild wheat, and these are the wheat which we are domesticated. And this is the corn which we had about 4,000 years ago in Peru. And through selection, we have achieved this one. And this is the wild tomato, and this is the tomato which we are using. So through selection pressure, we are causing genetic homogenization, and there is also loss of genetic diversity and some adaptive traits, which are important for resistance to diseases, for extreme conditions, and so on. So domestication has resulted in a genetic bottleneck in domesticated plants. So we need large genetic diversity or gene pool to tackle the current challenges, and crop-wide relatives provide untapped resources for genetic diversity. They have been through selective pressure as they have to go through several environmental conditions in nature. We have some crop-wide relatives which are adapted to drought conditions, and some of them are adapted to waterlogged areas. Others are adapted to warm temperature and cold temperature, and some others are adapted to saline soil, while the others are adapted to acidic soil. Some are resistant to insect and fungal pests as well as other diseases. And because of that, they have important genetic resources that are needed to cope with the changing globe and to feed the ever-growing population. And crop-wide relatives are wild plant species, which are closely related to our crops. By broad definition, it refers to all taxa within the same genus as a crop. And they are highly diverse. They account about 20% of the world flora. And currently, we are focusing on grassy, among others. And grassy belongs to the family Fabaceae. As you can see, its flower. And it can withstand extreme environment from drought to flooding. And it's usually cropped after the main cropping season. If crop fails, that means the same farmlands can be covered by grassy. And this makes it a climate-smart species. And the grassy is often the only alternative to starvation when other crops fail, especially in developed countries. It fixes nitrogen and serves as nitrogen fertilizer. Because of this ability to fix nitrogen, it can also grow on degraded land. It is very rich in protein, even as compared to other legumes. And it also serves as a fodder. And because of that, it's considered as a super crop to beat protein malnutrition in the future. The challenge is this plant presents a fascinating paradox. It is not only a lifesaver, but also it is a destroyer as well. It has a neurotoxin chemical, and when eaten as a large part for the diet for over a long period, it can cause permanent paralysis of the lower limbs in adults, and it can also cause brain damage in challenges. And this is often the case during farming periods when the other crops are failing. The opportunities, it is quite widely distributed throughout the globe, as you can see. We have diverse land races and farmers varieties. There are about 180 herbaceous species in the genus, which means that we have diverse wild relatives. It's herbaceous means it is easy to experiment with. And because of that, it has got a huge genetic resource. And certain varieties from Western Asia have been reported to have low neurotoxin. So what's our plan? Our plan is searching for varieties or wild relatives with low neurotoxin content. And this will allow to raise a grassy with reasonable neurotoxin content through breeding. We think that varieties growing in a similar ecotype most likely have similar characteristics and genetic resources. And because of that, we are thinking of using high resolution environmental variables such as mean annual precipitation, mean annual temperature, precipitation seasonality, temperature seasonality, precipitation of the wetter season, mean temperature of the wetter season, and a lot of other environmental variables. And by linking these environmental variables with the occurrence pointers, we can model the ecotypes. And so finally, we can link the ecotypes, the online available occurrence and the genetic databases with a digital twin. Building the digital twin model in a way it can make a report whenever new occurrence data and the genetic resources are put online from potential ecotypes or areas can be done. Especially the digital twin can notify us when data are updated from data poor areas or from poorly known habitats, and when the data are coming from marginal environments. So we use the digital twin service for automation of data flow, data fusion from distributed data sources from different data sources, both occurrence and genetic data, for dynamic model updating, feeding in updated environmental data followed by new model iteration, for automated model uncertainty analysis, that's comparison with the real life data, and for automated alert for new genetic diversity with predicted interesting target genetic properties or desired alleles. The benefits, this facilitates provision of vital information to plant breeders, conservation scientists, researchers, and the policymakers about the potential genetic resources from potential ecotypes, from data poor areas, from marginal habitats such as highly saline, acidic, dry, wet, warm, cold, and you can mention a lot. And this work can serve as a prototype for other crops as well, because similar method in the platform can be applied for other crops and crop relatives, wild relatives. The challenge to put the use case into practice are obtaining traits of interest through breeding is time-taking. Availability of whole genome data is really very important. And we don't also think the gene responsible for biosynthesis pathway of the toxin is mapped and well known. We don't have a lot of information on the inheritance property of the gene of interest. This, I would like to acknowledge Doug, who is the co-leader of this use case. I used a lot of slides from his previous presentation. The BioDT team for contributing ideas at Potsdam's workshop, especially Jeroen, who joined when we were discussing about this use case. That's, I thank you very much.
Thank you, Desalegn. I'm filling the break here between the speakers. My name is Dmitry Schigel, and together with Doug Anderson, I'm driving the work package of the Biodiversity Digital Twin project. In case you are in the wrong Zoom or just joined, or you still in the wrong Zoom, I want to remind you that this is the webinar, the second webinar of Biodiversity Digital Twin project, and today we are giving you some highlights on the use case work we have started a few months ago. We have hopped over the very important topic of what is biodiversity digital twin, for whom it is, and so on. Before the next speaker, I just very briefly want to remind you that the project website is available at biodt.eu, and you're welcome to explore the basics about the projects if you're wondering what's going on there. My version of our project reality is that we are putting together advanced modeling methods, supercomputers, and aggregated biodiversity and non-biodiversity data to build some standing multi-modeling solution, the dynamic one, that we hope will serve as a prototype of this kind of modeling work in the future. So it is the opposite of your familiar, let's build a model, publish the paper, and then try to write another grant. We're trying to build something dynamic and standing, and we're using 8 use case scenarios to pilot this. We're slightly afraid, but also excited, because probably nobody tried this before. And but in the use case line, we're very happy to have very energetic and knowledgeable use case leaders. And you heard from one of them. Thank you, Desalegn Chala, on presenting on the crop wild relatives. And the next speaker is Ingolf Kühn from Helmholtz Centre for Environmental Research, who will present another use case, this time on invasive and alien species. Thank you. It's all yours.
Thank you very much, Dmitry, for this introduction. Hello, everyone who's there. First, you may want to know what are biological invasions, or what are invasive alien species. And to go into this one probably has to know what are alien species at all, or alien or exotic species. And one can easily say, okay, they are those species that are not native to a region, you know, just by defining by the opposite. But this means they did not reach a region naturally after the last glaciation, but were introduced by humans either intentionally or unintentionally, or they made use of corridors that were established by humans, which facilitated their spread into new regions, for example, using canals or following roads or railroads or similar like that. And then a subgroup of these alien or exotic species are the invasive alien species. And there we have 2 different definitions. The one is the more scientific definition, that is, those are the species that spread rapidly. And for specific species groups, there are specific thresholds, what a rapid spread means, but they are different for different groups. Or, and that is the more political definition, or also the definition that was used in the Convention on Biological Diversity, is that they cause economic or ecological harm. You know, ecological harm is really that, for example, some of the species that you can see, like the upper yellow one is the goldenrod, that can grow in sensitive and rare dry grassland systems. Left to this is the black locust or Robinia, which is in symbiosis with specific nitrogen-fixing bacteria, and that pumps nitrogen into the soils of otherwise nitrogen-poor habitats, and this can cause ecological harm. Some of them can cause health harm. For example, ambrosia, which is extremely allergenic in their pollen, or the giant hogweed. When touching the giant hogweed and then being exposed to ultraviolet rays, it can cause severe skin irritations up to burn. And there are severe economic costs. And alone the cost of animal invasions in Europe was estimated to be more than $150 billion US, just from animals, as was focused in a recent special issue in Biota and Biological Invasions into these 2 journals. And the global costs of biological invasions are estimated to be at least $2.3 trillion US. So there is some real political demand and societal demand to counter these biological invasions, and therefore there's even a European Union regulation on invasive alien species there. The digital twin, we're going to focus on invasive alien plant species, because currently the data availability of this plant species is much better known than for very many other species. We do have some knowledge about birds and mammals or some insects, but they're usually less speciose. We want to model the level of invasions per habitat, which is how many species, how many plant species can be found in a specific habitat. What is the species pool of that habitat? And we know the habitat distribution across Europe, which means in principle we can provide a map such as the one that the colleagues 13 years ago published in Diversity and Distributions with the number of different alien invasive plant species in the different habitats occurring in different parts of Europe at a very high resolution, 1 square kilometer, for example. However, we can do that in a dynamic way, whereas the current map was just a static view of knowing habitat association and habitat availability, rather than really knowing where the species occur. We will then combine that with climatic suitability to know not only in which habitat the species occur, but also is the climate suitable for it, and taking into account changes of the past decades whenever possible from long-term observation projects, and then provide scenario projections and different management interventions, meaning if we change the management in specific habitats or the composition of specific habitats in the landscape, what does that mean for the distribution and the number of alien invasive species that are around there? The problem that we're dealing with is that we have very many different heterogeneous data sources. I just, sketched a one species matrix to the upper right. However, there is not just one species matrix. We have several different sources from where we take the species information. For example, for geographic breadth, we take GBIF data, among others. For temporal depth, we take the data from the European long-term environmental research and critical zone infrastructure that is being set up, and different other data sources. For these species, we do have habitat information. We have the habitat availability from different sources across Europe using different climatic input variables as models. And thus, we can not only model in which habitats how many species in principle occur, but knowing already how many species occur in which region and what the climatic requirements of these species are. We can combine all these different methods to come up with a probability surface. But because we have a very high resolution database, especially for habitat availability and climate availability, we have extremely large rectangular matrices, and to account for spatial dependence, we end up with extremely large distance matrices or spatial variance covariance matrices. However, that means we have reasonably good knowledge on spatial distributions, and by combining these different methods, we can come up with quite some knowledge on where species occur under which different climates, land management, and in the end also can translate this into spread scenarios and intervention scenarios. Now, the benefits of the project of the digital twin would be that we would unify the different decentral, very many different heterogeneous data sources that are there around Europe for this digital twin to get an almost real-time information on the different levels of invasions and the dynamics of invasions once the new data is entered into these different data sets. We can quantify the change, for example, what the different levels of invasions under different scenario projections mean as an increase in alien species in some parts of Europe and a decrease in alien species in other parts of Europe, but again, in a dynamic way, rather than the static that is just depicted there as a rough sketch. And we might even use that as an early warning system. However, the large benefit problem is probably that we can use these scenarios also to test for different interventions or management in habitats, as I just sketched out a couple of minutes ago. And we therefore really address policy requirements or societal requirements. And therefore, we think that this is a use case that is extremely relevant for decision makers and policymakers. Thank you very much.
Thank you, Ingolf. The second story out of three about how and where thematically we will be trying to build a prototype digital twin for biodiversity. I hope the audience is getting more clarity about why we're doing it. And thank you, Ingolf, especially for the closing slide where you stressed the need for science and policy, but also the right internal needs for our community working with biodiversity and non-biodiversity data. In spite of decades of efforts, we're still living in the world where data sources are largely disconnected, and we need to stitch them together. One way to try it is to put these data sources together for one big noble purpose, and we thought that biodiversity digital twin is such a thing. The third story in our sequence will be presented to you by Marcella Orwick Rydmark, and we move to the very exciting topic of pollinators. And out of 8 use cases, the species interactions use cases are maybe one of the more difficult ones in terms of data heterogeneity and so on. But we will hear from our presenter now what is the state and what are the plans for the use case. Thank you very much, Marcella, all yours.
All right, thank you, Dmitry. So I will be presenting today on behalf of the pollinator user case with the subtitle, A Digital Twin for Honeybee Dynamics in an Agricultural Landscape. So first, just briefly about pollinators. They're absolutely omnipresent in our ecosystem. So pollinators, we all know about bees, but as well as bees, there are beetles, there are butterflies, there are even small vertebrates that are extremely important as pollinators. So these vertebrates, for instance, can include lizards, they can include bats, they can even include small rodents. So pollinators are critical for food security and plant biodiversity. Now, with climate and environmental changes, the rate of acceleration of their decline is rapidly increasing. And I have to say that the full risks associated to this decline are not fully understood yet, but understanding these risks is absolutely vital to both science and to society. Now, just to briefly explain upon this, in this particular user case, we will begin to focus upon honeybees specifically as a pollinator, though I would like the audience to know that eventually this can be expanded to other pollinators. Now, honeybees, you may not think about this, but they are essentially domesticated livestock. So they have been bred for the pollination of our crops. And they're absolutely vital in this process and are estimated to be responsible for about 30 billion euros a year in ecosystem services in Europe alone. And if honeybees die, the crops that they pollinate will either die or be vastly reduced without massive efforts and massive costs by humans. So I've already discussed environmental and climatic changes for pollinators as stresses in particular, but for honeybees, there's a few other stressors that are also extremely well known. And so these can include mites, these include diseases, pesticides with specific classes in particular knowing to be extremely deleterious to them, modern agricultural practices that lead to forage gaps. And so in this case, a forage gap would be extended periods without any nectar supplies, and also modern day beekeeping practices. Now, no one single stressor alone will necessarily lead to a honeybee population decline. Multiple stressors simultaneously does lead to honeybee population declines. And what should be known and emphasized is that it's not practically possible to test these multiple stressors simultaneously with experiments. So instead, to understand honeybee population decline and honeybee dynamics, typically models are used. So in our case, the model that we're using is something called Behave. And so Behave has multiple modules in it for modeling different variables. So for instance, honeybee colony and population dynamics can be modeled. Foraging behavior of honeybees can be modeled. Landscape and forage availability. So for instance, different landscape usage scenarios and resulting forage availability for honeybees can be understood better. And as well, varroa and beekeeping stresses can be modeled within Behave. So as I mentioned, what's extremely important is the implementation of multiple stressors simultaneously. So within Behave, we can represent stressors within the hives. So this would include, for instance, varroa mites and diseases, as well as in the landscape. So nutrition, pesticides, forage gaps, and the weather. And this requires combining models of in-hive development, mite infestation and dynamics, as well as disease transmission. So BEHAVE already is a fairly sophisticated model. So what we want to know about is why BEHAVE would actually be interesting to actually implement BEHAVE within BioDT. Well, BioDT is essential for access to fast computing and comprehensive data expertise. It increases the model's spatial extent while keeping the resolution. So for instance, running the model for the entirety of Germany and comprehensive uncertainty and sensitivity analysis. Now, as well as we've previously emphasized, BioDT is a prototype, and BEHAVE is already a model that we know quite a bit about and we can already work with. And there's also quite a lot of data already available for honeybees. And so BEHAVE currently is a model. So how do we turn this into a digital twin? Well, there's several things that are necessary, but for instance, automation of the data flow, dynamic model updating. So for instance, the feeding in of updated environmental data followed by a model iteration. And we could imagine this happening, for instance, one time a year at that level of granularity to begin with, perhaps even more regularly though in the future. And automated model uncertainty analyses. So comparisons with real life data, which we have a lot of. As well, there are quite some outputs that we could actually envisage when implementing BEHAVE within BioDT. So right here, I actually am showing a drought risk assessment map, but we could imagine that we could make similar maps for that are interactive and or static, where the color could code the risk of forage gaps and hence honeybee colony failure. And so this will indicate where certain changes in land use could and should improve the forage supply situation for our pollinators. As well, we could think about something called ecological focus. And so this is a greening solution within Europe. And right here, I'm showing a few different cases where, for instance, we could actually model the introduction of different nectar sources within crops to reduce forage gaps. So in the first example that's modeled here are 4 different crop types without any introduction of other nectar sources, where there's several forage gaps that are present. In the next example, we have introduction of nitrogen-fixing clover cultures, which does a pretty good job of actually reducing forage gaps. And this has already been shown experimentally to have more or less these sorts of effects. So the model does agree with reality in this case. And in the third case right here, we're actually showing crops that are introduced with 10% flower strip margins. So this is actually not very commonly done within the farming community, but does suggest, for instance, that if you were to compare with a clover culture co-introduction, that actually introducing these different flower strips might be even better for reducing these forage gaps. And so who would actually be using BEHAVE within BioDT? Well, we can think of academia. We can think of beekeepers and also their institutes and associations that they work with, farmers and land use management, as well as policymakers in land use and legislation. And so finally, I want to make some acknowledgements here. So Professor Volker Grimm is the user case 4 leader, and he provided me with many of the slides that I'm presenting today. And Volker will, within next year, be joined by Jürgen Grunewald. Both of them are based at the Helmholtz Centre for Environmental Research. And as well, I would like to acknowledge BEHAVE and the team that developed it. So this was a team of researchers within both Germany and in the UK, and they already as well had some funding from industry as well as from the Biotechnology and Biological Sciences Research Council. And so thank you very much.
Thank you, Marcella, for your last presentation. And I would like to thank again all our speakers today. And I hope that all people that are attending today could also understand a little bit better what is the scope of our use cases, and also see that actually they're not oriented for research and academia, let's say, but also other stakeholders can use it, like as Marcella said at the very end, all the work they were doing can be also useful for policymakers, and for example, also be used for addressing some important topics like the one that are addressed for the in the European Biodiversity Strategy to 2030, for example. Now we will move to the last part of our webinar, let's say, where we will actually give the opportunity to you all to ask us any questions. And before doing that, actually, I would like to still, let's say, your time for other 5 minutes for doing a last, let's say, Mentimeter quiz together. So I will share again my screen with another, let's say, set of questions from Mentimeter. The way of working is exactly the same. So what you have to do is go through menti.com and then use the code that you find here. So 58242641. And we can also copy the link here in the chat so that you can also see this. And so the first question, that it is a little bit more technical compared to the first Menti question. So here we're curious to know if you have already worked with some data that are used by our use cases, and for which purpose. Okay, so it seems that they're very innovative, the use cases that we're developing. As of now, okay, they are very new. And okay, we got actually just some comments, let's say, either for previous projects or let's say previous analysis that were done. Okay, they were mainly used okay also for modeling. And okay, I see that someone has also used the GBIF let's say database, which is also used by our use cases, as Desalegn was also mentioning. And okay, also for Earth observation. Okay, thank you. And I will move then to another question. If let's say, following the presentation that we have done, if you can really, let's say, summarize with maximum 2 words, you can also use one. What are the main benefits that are brought by the BioDT use cases? And if you could really, let's say, highlight something from the interesting presentation that we heard so far. Okay, so we have up-to-date production, which is good, data access and integration. Then we have, okay, also pointing to the open service, which is, I mean, also very important, considering also the strategy, let's say, the research strategy that we have now of making knowledge, let's say, accessible openly to everyone. Then, okay, we have again the policy topic, also sharing of knowledge, creating synergies, which is also something very important for European project and also for us, as we're also looking for new opportunities to learn from other projects and understand how we can better also do our work and do our model. Then, okay, we have, okay, also another point about knowledge, about okay, also food security. So we have also some points linked to the to some use cases that we have touched today. This one is most probably a little bit more technical as question. So in case some of you have already some experience in building some digital twin models, did you experience some issues? And if you have, actually, it would be nice also to hear from you. Okay, which is very nice, then where digital twin is. Okay, so I see that the most difficult thing is really about understanding how to build the model, which is, yeah, not very easy. Then, okay, also another point is about the complexity of biodiversity, because it is so wide that it is very difficult to cover with use cases. Yeah, for example, also in our case, we have let's say we have selected 8 use cases, but obviously the scope is wider. Also, okay, another point is about obtaining comparable data between different data sets. Yeah, because also here we should use quite a lot of standard semantic rules for making them interoperable, even though this isn't always an easy task. Also, again, I find other points about the data heterogeneity, the standardization, as we said. So it seems, I mean, that it is very hard to connect different data together. So this is the one of the biggest difficulties that the team finds. And the last one you said, it is, I mean, for you also to understand, let's say, for us to understand, what are you expecting actually from the biodiversity digital twin, which may be also the reason why you have also joined our webinar today. It could be, I don't know, about learning something more about the use cases, or defining a little bit better what is the biodiversity digital twin. As in the previous question, we got quite a lot of comments also that people are not really clear about what a digital twin is. Okay, we got actually 2 interesting comments so far. So people would like also to understand how to improve all the integration between data, then also, let's say, improve the point about data accessibility, and then, okay, here we have also some points about creating something that is also linked to lakes and rivers, and then to develop real BioDT models. Okay, other comments actually about the data integration, and also, let's say, deliver, also say something reliable that then can be used also for additional research. So I would like to thank you again for your time and replies to this question. And now instead, I will pass the floor again to Dmitry, who will instead moderate the Q&A session with all of you.
Thank you. Yes, looking at the questions and answers coming, and a very difficult one, I thought that, whoa, this is the scariest Q&A session that I had ever. And but also looking at the answers coming through mentoring exercise, I realized that many of you appreciate the difficulty, novelty, and the complexity of the tasks that we bravely took. Thank you for the questions. First of all, we have received 16 questions, and 11 of them, they were answered while we were collecting them, but 5 of them remain open. So I might start with them, and I rely on my co-panelists to help me answer them. A couple of questions from Irji Klik on Lumi supercomputer and the heavy acronyms about some computer architecture. I have to confess that I know nothing about what you ask, but we rely on the Lumi supercomputer. First of all, our supercomputer partners in Finland are eager to make this infrastructure. Recently launched, busy with some very heavy computational tasks, and they can be heavy in terms of calculations. They can be heavy in terms of how much data can be fed simultaneously and so on. But I think, Irji, you might need to wait for another webinar or another opportunity to get your technical questions answered, or reach out to the BioDT project people, including Doug and myself. We will connect you to the work package 3, very knowledgeable people who are running all the computational things. Maybe, Doug, you can save me out of this. Do you want to add something?
So I also believe that we have to rewrite some of the code. I don't know in detail these libraries that are mentioned by Jerry. But we do expect that some engineering of models and code and adopting it. There somebody talking? Yeah. No, so I agree. We will probably refer to project partners for the details.
Okay, then there is a question from John A.B. about DT framework or services that scale better as regard data are not very much available. John, you reveal one of the key motivations for the research infrastructures to join the BioDT projects, that data availability, in spite of the very big numbers we're all proud of, remains an issue. And the digital biodiversity data and the non-biodiversity data we have for the planet, it's not evenly covering the planet, and it's not evenly covering time periods, of course, and different taxa and different themes, that some of which we presented to you. So experimenting with biodiversity digital twin is, of course, an attractive task in itself, but for many of us joining this project, this is an opportunity to connect the data and scale it up to the limits we haven't attempted to reach before. So the framework is, we have 8 use case stories, basically thematic spaces to experiment with different, not necessarily connected modeling approaches, and then see the data availability and the data gaps that are the reality today for these use cases. Then we will attempt to stitch it all together to see if we can improve the data coverage, or maybe we can compensate for the lack of the data or the shortcomings of the data sets with modeling. There will be a gradient of broad and shallow, or narrow and deep use cases and the use case stories there. So I hope these things will help us to improve the data, basically. Then there is a question from Christos, to what extent data generated from modeling can be inserted to the subsequent analysis? This is a very good and deep question, and we have seen already in the use case building that some of the input data into the BioDT models is not primary, it's actually modeled. I would like to ask my panelists today to unmute and add something on this topic if you have additional comments, please.
Yeah, and I'm also sure that Christos will help to design this workflow. So the components of the twin will be in a Galaxy workflow so that you can chain them together in subsequent analysis and take pieces of them. So I'm sure that Christos will help us to build this vision to make it more reusable and available on component level.
Thanks, Doug. Of the questions we haven't touched yet, there is one from Joe Jordan. If data covering a large enough spatial area with sufficient measurement density is not available, what plans, if any, are there to generate synthetic data to all work to proceed in parallel and speeding up the models and underlying libraries? There is a lot of discussion about what shall we do if the data is not enough. Every use case leader is very sharply concerned about such situations. So there are several solutions for this. One of them is indeed the synthetic data or the play data. And I'm sure during the modeling experiment, some of this approach will be put in place. From the perspective of the data availability, we plan to start simple. We plan to start from the data stories where data is easily available now. Because we're building the prototype, it's more important for us to show that it can be done in principle. We're piloting. We don't attempt to cover the complexity of biodiversity, to cover the whole planet with all possible methods and modeling algorithms. I don't think it's possible ever, but it's fun to try with what we have with the 8 use stories with synthetic data, I'm sure. Then there is a question for Desalegn about DNA-based observations that have no taxonomic classification, the OTUs and ASVs in calculation of biodiversity metrics. Would you like to answer this, Desalegn?
Sorry, what the question again?
The question is, are you planning to include DNA-based observations that don't have taxonomic classification? So basically, when the evidence of something present in time and space, the only thing you know is the sequence, and you cannot assign it to a known taxon, but you can maybe classify it to a certain operational taxonomic unit or species hypothesis. Do you plan to do that in the crop wild relatives context?
Yeah, actually, Doug may comment on it later on. Actually, what we are planning to do is we are just planning to model the ecotypes. And then we are also planning to check what kind of genetic data is coming from each of the ecotypes and which ecotype are lacking genetic data and so on. So finally, we are also planning to link the digital twin with online genetic databases so that the digital twin can alert us whenever new data is updating. So we are using it for some practical reason. And then when we are not sure of the taxonomic unit and so on, there is a level of uncertainty which you can entertain. So we'll be careful about that. So maybe Doug may add on this.
Yeah, and also we look at Latteris, we look at the particular genus, so it's known to us. So we are not trying to look at completely unknown, unidentified OTUs. I don't think that would make sense in this use case, but there could be other use cases.
Yes, there is a sister use case to this under the genetically detected biodiversity, where we will attempt to model patterns and processes for poorly known habitats, such as for instance, soil. And anybody working with soil biodiversity knows that most of the fungi and a lot of invertebrates, they don't have valid scientific names, but they are repeatedly found in the samples because you know them from eDNA, you know what they cluster with. So the OTU approach will be probably very present there. To stay a tiny bit more on this question, I think it would be important to stress that differently from many other use cases, the agrobiodiversity related one is likely to handle taxonomic levels below species. This is maybe something not very familiar for people working on invasive alien species or threatened species. Infra-specific categories, they are not very much respected in the fundamental biodiversity research, but they are essential in agrobiodiversity, and this brings an additional access of complexity and the challenge to us to handle that.
You can add that some of the crops are hybrids between species, so they don't have clean taxonomic anchoring.
Yes. There is a question from Arildo Diaz asking that there is not enough information on the use cases. And exactly this morning, when we had a catch-up call for the use case leaders, somebody suggested that we should very soon bring more clarity on what's going on with this use case building. I would like to highlight that we are in the beginning of the project. The use case leaders, many of them were appointed yesterday night to start on this, some of them only a few weeks ago. I would like to give people some building and writing freedom and peace for a month or so. And there'll be a workshop in the project in November in Finland, where the use cases will be presented to the modeling work package. After that, I would say second half of November, we'll feel much more comfortable and ready to reveal some of the use case work to the broader community through the website. We have 1 minute left. I would like to ask Rita if we need to do something else, or we will.
No, we're actually at the very end of the webinar, so we can close it right now.
Okay, so some thanks in the Q&A. Somebody has asked what EBV stands for. It's Essential Biodiversity Variable. You can Google the rest. Thank you very much for coming. It was great to have so many people interested in our use cases, and we hope to tell you more stories in the future. Thank you all.